In this video, we are going to address one of the most intriguing topics of biochemistry, that is the enzyme inhibition kinetics. Now, before we move on to the enzyme inhibition kinetics, we need to know something which is called the Michaelis Menten equation. Michaelis Menten equation. Michaelis Menten equation, what it says that the velocity of the reaction is equal to the maximum velocity of the reaction multiplied by the substrate concentration divided by the Michaelis constant plus the substrate concentration. So this is the Michaelis Menten equation. Now, Km, the Michaelis constant, it is that substrate concentration. When the half of the maximum velocity is reached. Okay, so Km is that substrate concentration when the maximum velocity is half of the total. Now we can draw this out in a graph where the x-axis would be the substrate concentration and the y-axis would be the velocity of the reaction, and we can plot an enzyme kinetics like this. Now there would be one part of the reaction where, at the end, when all the enzymes present in the reaction is saturated with substrate and no more enzyme substrate can bind. That is why the graph tends to flatten out at the end. And this point of the y-axis is the maximum velocity, or Vmax. We take the half of Vmax and extrapolate it to the x-axis. We get the value of Km. This is Vmax by 2. So this is Vmax by 2, this is Vmax, and the point where it hits the x-axis is Km. Now this Michaelis Menten equation, this can be tweaked a little and made into a linear graph format. So how do we do that? We reciprocate the equation. 1 by V naught is equal to Km plus the substrate concentration. We are reversing the order by Vmax into substrate concentration. Okay, now we sort of separate them out, and 1 by V naught equals to Km by Vmax into 1 by substrate concentration, plus, if we separate the other half out, we'd get 1 by Vmax. Right. So this equation is called a double reciprocal equation, or the graph that is going to be generated is called the Lineweaver-Burk plot. Now, how does that look? We draw it like this. The y-axis would have 1 by V naught, because if you notice this equation, it is in the form of y equals to mx plus c. Y equals to mx plus c. So this is my y-intercept, this is my x-axis, and this is my slope, Km by Vmax. And we draw that out in the graph. We get a linear sort of graph. So where the line hits the y-axis, this region, this is the y-intercept, or 1 by Vmax. The slope is m, which is Km by Vmax, and the line where it hits the x-axis, this is negative of 1 by Km. Now everything is reciprocated, so, and since this is in the negative axis, so it is negative 1 by Km. Now we can use these two formats or these two graphs to understand the enzyme inhibition kinetics as well. But first of all, let's look into the various types of enzyme inhibition kinetics or the enzyme inhibition that occurs. So the first type is called the competitive inhibition. Competitive inhibition. In competitive inhibition, what happens is that we have our enzyme, and let's say this is the active site. The substrate, let's say this is the substrate, and the inhibitor, they compete for the same site or the same active site to bind to. This would also have a binding site over here. This would also have a binding site at the same place. So they're competing against each other to get the enzyme. Now, if that happens, if the substrate and the inhibitor, they compete for the same site, what we can do, we can increase the substrate concentration. We can increase the substrate concentration so that this concentration, the inhibitor concentration, is negligible. And once we do that, we would eventually get the probability of the substrate binding to the enzyme much more than the inhibitor, because the concentration or the amount of substrate is much more. So to deal with this kind of inhibition, we increase the substrate concentration. And how would the graph look like? Now, let's say, again, we start off with the Michaelis Menten plot, where we have substrate concentration on the x-axis and the velocity on the y-axis. So normally, without the inhibitor, we would have a normal inhibition enzyme kinetic, and where the Vmax is over here. And when the substrate, when the inhibitor is added to the solution, what would happen? The Km, where the substrate concentration, at which the velocity is half of the maximum velocity, that would increase, because some of the inhibitor is binding to the enzyme, and to get rid of that, we are increasing the substrate concentration. So the inhibition would look something like this. And if this was the Vmax by 2, the half of the maximum velocity, and this was the Km, this was the Km for the normal enzyme kinetics, then, like I said, the Km would increase, and the Km would come somewhere over here. But one thing is that the Vmax won't change. The Vmax would remain the same, because when we are increasing the substrate concentration, eventually the same old Vmax would be reached. But what happens is that more substrate is required. So, like, more substrate is required. That is why the Km, or the substrate concentration at which the maximum velocity, or the half of the maximum velocity is reached, that increases. But the Vmax would remain same. Eventually, the Vmax that was there for the normal kinetics would be reached. And if we put this graph in a Lineweaver-Burk plot, or the linear plot, we would have something like this. Let's say this is the normal kinetics. Okay, we have the 1 by V naught over here. We have the substrate concentration of 1 by S concentration over here. Then this region would be 1 by V max as usual, and this would be minus 1 by Km. This is for the normal without the inhibitor. Now when the inhibitor is introduced, what happens? Like I said, the Km would increase. Now as the Km would increase, this whole value, since it is negative, this would shift towards the positive, because increasing the Km or increasing the denominator over here, that sort of lowers down the negativity of the whole number. So the overall number, it gets pushed towards the positive side. So we have something like this. Now the Vmax would remain the same. Like I said, the Vmax point on the y-axis, or 1 by Vmax in this case, it would remain the same. But what happens is that the 1 by Km, that shifts towards the right, and the Km increases by a factor of, say, alpha. Okay? So when the inhibitor is present, the Vmax remains same, but the Km increases, and since the denominator over here increases, it shifts the overall value, that is negative, towards a little bit positive. Now, this is the two plots for the competitive inhibition. Now, moving on to the non-competitive inhibition, the second type, which is the non-competitive inhibition. In non-competitive inhibition, what happens if this is the enzyme, let's say this is the enzyme, the substrate and the inhibitor, they have different binding sites. Okay, so if the substrate is binding over here, the inhibitor has a different binding site, which is also called the allosteric site. The inhibitor would bind over here, and this is the substrate. And the inhibitor can bind when the enzyme is free of the substrate, or it can also bind when the substrate is bound to the enzyme. So in non-competitive inhibition, this can be the case, this can be case 1, and the inhibitor can also bind if the substrate is not there at all. If the substrate is not there at all, then also the inhibitor can bind. These two are the situations for non-competitive inhibition. Now, let's say we increase the substrate concentration. Will that affect the enzyme or the kinetics of the enzyme? No, because even though we increase the substrate in the solution, the enzyme would bind to the inhibitor, and that would sort of inhibit or lower down the activity of the enzyme. Now, if we draw the curve of this, let's draw the Michaelis Menten curve first. We have we have V naught and the substrate concentration. The normal kinetics would be the same. Where this is Vmax and this would be, say, Km. But when the inhibitor is present, what happens? The Vmax reduces. The Vmax reduces. The Vmax reduces because the inhibitor is not binding to the same position where the substrate is binding, or the active site, but it is binding to a different position. And it does not matter whether we increase the substrate concentration. The inhibitor would eventually bind to the allosteric site and sort of reduce the rate of the reaction. And that is why the rate of the reaction decreases and a new Vmax is achieved. Right? But one thing happens is that although the Vmax is lowered, since the substrate concentration is not changed, the Km remains the same. The Km would be the same. The Km does not change. Only the Vmax changes. Right? So this would be Vmax by 2 for the normal one, and this would be Vmax by 2 for the inhibition. So once more, if we give the inhibitor inside the solution, it does not matter we increase the substrate concentration, because eventually the inhibitor would bind even if the substrate is not present. And since the inhibitor would bind to the enzyme, the increase in the substrate concentration would not matter, but the Vmax would eventually decrease because the inhibitor is binding to the enzyme even in the absence of the substrate. So we keep the substrate constant, but the Vmax decreases and the Vmax by 2 decreases, or the half of the Vmax decreases, but the Km, since the substrate concentration does not affect the inhibition, the Km remains constant. Now, if we draw this out in a Lineweaver-Burke plot, what happens? So this is 1 by V naught, and the x-axis is 1 by substrate concentration. Now the first is the normal one, where 1 by Vmax is this point. Now when we give the inhibitor and we draw it in this Lineweaver-Burke plot, what happens, like I said, the Vmax decreases, the Vmax comes down. So the overall 1 by Vmax should increase. But the Km remains constant. The Michaelis constant is the same for both for both without the inhibitor and with the inhibitor. So the Km, if this was my minus 1 by Km, this would remain constant for both the thing, but the Vmax that would decrease, and hence in this plot 1 by Vmax would increase. So this would be 1 by Vmax, and 1 by Vmax is increasing by a factor, let's say alpha dashed. Okay, so in the previous one, what we saw is that the Km is increasing by a factor of alpha. But in this one, what we are seeing that the Vmax is decreasing by a factor of the alpha dash, whereas the Km remains constant. Or the Km remains same for both the stuff. Next, the third one and the final one, we have something called the uncompetitive inhibition. Uncompetitive inhibition. Now, for uncompetitive inhibition, let's say this is the enzyme. This is the enzyme, and every time I draw the enzyme, I forget to draw the allosteric site. and this is the allosteric site, what happens is that the substrate would bind to the enzyme, and only in this substrate bound state, or the state which is called the enzyme substrate complex, only in this ES state the inhibitor would bind. When the substrate is not there, the inhibitor cannot bind. But when the substrate is there, the inhibitor can bind. Now, this is the difference from non-competitive inhibition. Because non-competitive inhibition, we were seeing that the inhibitor can bind either in the presence or in the absence of the substrate. But in uncompetitive inhibition, the inhibitor can only bind when the substrate is bound to the enzyme. Now, how would the curve look like? Now, one thing, this kind of situation can be dealt by decreasing the substrate concentration, because the more substrate is there in the solution, the more it will bind to the enzyme, and that would trigger the inhibitor to also bind to the enzyme. So we can decrease the substrate concentration to deal with this kind of inhibition. So decreasing the substrate concentration, because more substrate will cause more enzyme complex to form, and that will increase the inhibitor binding. Therefore, if the substrate is decreased, the original Vmax would never be reached, and a new Vmax would be formed. So let's see that in the graph. Now, if we draw the normal without the inhibitor, if we draw the normal graph without the inhibitor, it would be the same. And this would be the Vmax. And we have the V naught in the y-axis, substrate concentration in the x-axis. And when we plot the kinetics of the inhibition, what would happen is that it would be same like the non-competitive one, where the Vmax decreases. But another thing would happen, the Km would also decrease, because to deal with this situation, like I said, we have to decrease the substrate concentration. And since the substrate concentration is decreasing, the Kmax eventually would decrease. If this was the Km, then in the inhibition one, the Km would decrease somewhere over here. In non-competitive inhibition, only the Vmax was decreasing. The Km was the same for both the curves. Now, for the uncompetitive inhibition, the Km is also decreasing, because we are decreasing the substrate concentration. But for this one, we were not decreasing the substrate concentration, because decreasing the substrate concentration would not have any effect, because it was either way binding to the enzyme. The inhibitor was either way binding to the enzyme. So now let's put this into the Lineweaver-Burke plot. We can have 1 by V naught over here, 1 by substrate concentration over here, and the normal plot would be the same. But in the inhibition one, and the normal plot would have the 1 by Vmax over here. In the inhibition one, the Km is also reducing, as well as the Vmax. Now, if the Vmax is reducing, then if the Vmax over here is reducing, then 1 by Vmax as a whole, that should increase. Just like the non-competitive one, 1 by Vmax should increase. So it should go somewhere over here. And we had the minus 1 by Km over here, this point. Now, the Km is also decreasing in this case. So if the Km is decreasing, so the overall negative 1 by Km, that should also be more negative, or that should also shift towards the left. So the whole value should shift towards the left because Km is decreasing. Km is decreasing. That's why minus 1 by Km should shift towards the left because more negative value is being imparted on 1 by Km. Right? Over here, the Km was constant, so it was at the same place. But for this one, the Km, since it is decreasing, the overall 1 by Km or negative 1 by Km, the negative value is increasing. So if you're increasing the negative value, it should move towards this side. Then it would be somewhere over here. And now if we draw the curve, it would be parallel to the original curve without the inhibitor. So now we have decreased the Km, let's say by a factor of alpha dashed Km minus alpha dashed Km, this point. And we have also decreased the Vmax, thereby increased the value of 1 by Vmax by a factor of alpha dashed. Right? So once more, in uncompetitive inhibition, the inhibitor can only bind if the substrate is bound to the enzyme. Now that leaves us with only one solution to deal with this situation. We have to decrease the substrate concentration because increasing the substrate concentration that would help the inhibitor to bind to the enzyme because it can only bind when the substrate is present. So we are decreasing the substrate, and since we're decreasing the substrate, the Km value is decreasing, and since the substrate is getting lowered, the original Vmax or the maximum velocity can never be reached because the substrate is less, and that is why the Vmax value also decreases. Putting that into the Lineweaver-Burke plot, the Vmax value decreases, and hence the overall 1 by Vmax increases by a factor of alpha dashed. That is why it is above this curve. And since we are also decreasing the Km or the Michaelis Menten constant, that is why the Km decreases and the overall negativity of minus 1 by Km increases. So the negativity increases. Now it becomes more negative. Since it becomes more negative, it shifts towards the negative end of the graph. And this shift is also, let's say, by a factor of alpha dashed. So we get a new Km, which is alpha dashed by Km. So that was all about the competitive, non-competitive, and uncompetitive inhibition.